If there aren't any questions, let's begin. Let's see. Got the red dot there. Can somebody confirm you can hear me?
I can hear you.
Great, thank you. All right, so we were talking about photosynthesis, and we mentioned there are two types of photosynthesis: cyclic photosynthesis and non-cyclic photosynthesis, which most of you are probably familiar with non-cyclic photosynthesis, where 6 CO2, 12 water, and light makes glucose, and 6 oxygen and 6 water. In cyclic photosynthesis, ATP is made. In both cyclic photosynthesis and non-cyclic photosynthesis can be made in every photosynthetic cell that I know of. And I don't know of every photosynthetic cell, but in green plants and green bacteria, meaning photosynthetic bacteria, all cells that I know of can engage in both types of photosynthesis. They use cyclic photosynthesis when they want to make ATP. They use non-cyclic photosynthesis when they want to make glucose. Let's talk a little bit more about cyclic photosynthesis or cyclic photophosphorylation. Light starts the process, and the light hits the electrons in chlorophyll, and the chlorophyll is in the protein, the photosystem 1, and the light causes those electrons to move, meaning the light energy gets absorbed by the electrons, and that excites the electrons, and then they move. The electrons move to this protein in a membrane, and to this protein in a membrane, and to this protein in a membrane, and back to this protein in the membrane. And you'll notice that the electron starts from chlorophyll and it ends up at chlorophyll. Why it's called cyclic photosynthesis or cyclic photophosphorylation. Phosphorylation is where you add a phosphate group to a molecule, and as the electron is moving through this protein in the membrane, it gives this protein the energy it needs to transport hydrogen atoms from this side of the membrane to that side of the membrane. That creates a twofold gradient, and that is there's more hydrogen on this side of the membrane than that side, and then there's more positive charges on this side of the membrane than that side. The hydrogen ions will want to move to this side of the membrane by the laws of diffusion, as well as, you could say, by the laws of electrostatic attraction. Sorry, I've got a frog in my throat. The only place the hydrogen ions can move across the membrane is at this protein here, or this channel protein, which is an ATP synthase enzyme. And when the hydrogen ions flow through this protein, it gives the protein the energy it needs to phosphorylate ADP to make ATP. Any questions about that? The mechanism for making ATP using this method is very similar to the way that aerobic respiration makes ATP in the electron transfer chain. So this process should be familiar to you. You don't need to know this, but This membrane is in a chloroplast, in a plant cell, or an algae, and it's the cell membrane of a prokaryotic cell. All right, any questions about that? The last slide I'll let you read over. It's talking about a summary of ATP production. Making ATP, the energy source can vary. The electron carriers can vary. And then the final electron acceptor can vary. Any questions about Chapter 5? If not, I want to quickly start Chapter 6 because we have to finish this chapter today. Sorry, I meant to show you this. For Chapter 6, we only have one day on the schedule. That's today. We do have a lab today. So, like always, I start a chapter giving you the major goals and the rough outline. Know the terms for this chapter. Understand the various growth requirements. So what requirements there are for microbes to grow. Understand the 4 phases of growth and be able to compare the different methods for measuring microbial growth. Any questions about any of that? When we're talking about microbial growth, you should understand that we're talking about an increase in the number of cells, not an increase in the cell size. And this is a little video that will show you microbial growth. You can see why.
Bacteria reproduce very simply and rapidly by doubling their contents and splitting in 2. Just one bacterium, dividing every 20 minutes, could produce nearly 5,000 billion bacteria in one day.
Okay, any questions about that? There are requirements for our microbial growth. There are physical requirements and chemical requirements. Physical requirements mean that the microbe has to be at the correct temperature, the correct pH, the correct osmotic pressure, usually meaning the correct salinity, etc. The various chemical requirements for growth is that microbes have to have a carbon source, a nitrogen source, a sulfur source, a phosphorus source. They have to have trace elements available, and the correct oxygen and organic growth factors. Any question about any of that? When we're talking about temperature, each microbe has its own minimum temperature, maximum temperature, and optimum temperature that it'll grow at. You can classify microorganisms into groups on the basis of their preferred temperature range. Psychrophiles, or organisms that like around 10 degrees Celsius to grow. These would be the microorganisms that grow within your refrigerator. And they cannot infect us because our temperature is around 37 degrees Celsius, and their cells would fall apart at their temperature. At least their enzymes would take on the wrong folding pattern and conformation pattern, and their enzymes probably would not function. Psychotrophs are things that grow best around room temperature, so around 22 degrees, 21 degrees. And those would be like organisms you find in the soil or on your countertop. mesophiles are organisms that grow best around 37 degrees Celsius. All human pathogens are mesophiles, and that's because mesophiles grow at our body temperature. And so an organism that grows at our body temperature would be or could be a human pathogen, and that's the temperature that all human pathogens grow at. Thermophiles are organisms that grow at elevated temperatures, 60 to 65 degrees. And then a hypothermophile is an organism that was not known when I took microbiology. It's an organism that prefers to grow, 90 to 100 degrees Celsius. Now you should realize that the exact range and even the optimum temperature for a species is not rigidly defined, meaning when we're talking about mesophiles, the exact range isn't 10 degrees to, 49 degrees, and that not all mesophiles grow at an optimal temperature of 37. For example, M. luteus is a mesophile, but it grows best at 30 degrees Celsius. And the reason for that is that it grows on our skin. And our skin is a little bit cooler than our internal body temperature. So M. luteus is a mesophile, but it doesn't grow best at 37. It actually grows best at 30 degrees. Any question about the different temperature classifications and how the range as well as the optimum is not rigidly defined? Okay, I didn't hear anything, so we're going to move on. Food spoilage can occur when food is left between, something like 15 degrees, maybe 17 degrees, to 51 degrees Celsius. And that is when bacteria can rapidly grow. And so food that is left at that temperature for 4 hours, unless it has a preservative in it like sugar or salt, it should be thrown out after 4 hours because the bacteria can rapidly grow and spoil the food. Bacteria can grow at temperatures below 15 degrees and above, 51 degrees, but that bacteria grows slower. And as you know, bacteria can grow in food in your refrigerator, but the bacteria grows slow and the spoilage of the food can happen slowly. So you should throw that food out after a week in the refrigerator, unless the food has a preservative like salt or a lot of sugar or an acid like ketchup, for example, lasts a long time in the refrigerator, and I'm going to say at least 2 years. Okay, any question about any of that? Going to skip the next slide about food spoilage. And actually we're going to start skipping these slides because the physical requirements, pH, and the next slides are all topics that you will cover in your worksheet for the lecture. And because I've got to get through this lesson today, I'm going to skip these slides. Any question about any of that? You do need to know the material, but you'll be getting it in the in the worksheet for this lesson for the lecture. All right, so it ends in the 4 toxic forms of oxygen. And so the next slide I'm going to talk about is some chemical requirements that all organisms, especially microbes, need. And one of them is oxygen. Microbes that use oxygen are called aerobes. And aerobes produce more ATP from their nutrients than microbes that do not use oxygen. And those we call anaerobes. And the reason for that is aerobic respiration is the most efficient way for generating ATP. And so aerobes produce more ATP from a molecule of glucose than an anaerobe. You can classify microbes on the basis that oxygen has on their growth. Let me blow this slide up. There we go. So we can make a thioglycolate tube, and when that tube comes out of the autoclave, thioglycolate removes all oxygen from the media. However, the tube is open to the air, and so oxygen comes back into the tube. And so at the top of the tube, we'll have about 21% oxygen. And as you go down the tube, oops, didn't mean to do that. As you go down the tube, you'll have less and less oxygen until you come to the bottom of the tube where there will be essentially no oxygen. So a thioglycolate tube creates an environment where there's a gradient of oxygen going from the air at about 21% at the top of the tube, and then the oxygen decreases down to 0% in the bottom of the tube. Any question about any of that? So we can inoculate the tubes with bacteria species and then look at where the bacteria grow. If the bacteria only grow at the bottom of the tube, and as you go up in the tube you get less and less growth because there's more and more oxygen, we can say that that organism is an obligate anaerobe. The bacteria only grow where there is no oxygen. Any question about that? On the other hand, if the organism only grows at the top of the tube, where you have the most growth, and then as you go down the tube, you have less and less growth, which that's trying to show you there. They should put a 1 dot right there. Then we can say that organism is an obligate aerobe. It only grows where there is oxygen, and as you go down in the tube, you get less and less growth because there's less and less oxygen. Now that's different from a facultative anaerobe. These organisms are anaerobes, and they will grow everywhere in the tube because they do not need oxygen to grow. But they are facultative anaerobes, and they will grow best at the top of the tube where the organism is performing aerobic respiration, making the most ATP. And that's why you see the most growth at the top of the tube. But as you go down in the tube, and that's not shown very well, as you go down in the tube, you'll get less and less growth, but you will get growth everywhere in the tube. Okay, it just will be less growth than high up in the tube. And that's because these organisms down here are not performing aerobic respiration. They are performing either some other mechanism of respiration or fermentation to grow, to make ATP. There's another anaerobe, and these we call aerotolerant anaerobes. These organisms do not need oxygen to grow. They do not use oxygen, so they can grow everywhere in the tube. And they have the enzymes they need to detoxify the toxic oxygen metabolites. And that would be things like free oxygen radicals. So aerotolerant anaerobes grow everywhere in the tube uniformly, meaning you'll see the same growth at the top as in the middle and the bottom of the tube. Any question about aerotolerant anaerobes? All right, no question. The last one is a microaerophile. These organisms are aerobes. They do perform aerobic respiration, and they have to perform aerobic respiration, so they do require oxygen to grow, but they do not like full oxygen. Come on, mouse. So you'll not see growth at the top of the tube. You'll see the best growth somewhere in the middle of the tube, where there's less oxygen than 21% oxygen. And as you go down the tube, as well as up the tube, you'll see less and less growth. These organisms like growing at reduced oxygen concentrations, but they do require oxygen to grow. An example of that is Helicobacter pylori, an organism that grows in your stomach. And it requires oxygen to grow, but it doesn't like to grow in 21% oxygen. It grows in less, something like 4 to 10% oxygen. And why your stomach is less oxygen than 21% is because the microbes in your stomach are using up the oxygen, so the oxygen decreases in concentration, but the oxygen is always present because the stomach is open, like when you swallow, to the air. Any question about any of that? All right, if not, let's move on and talk a little bit about culture media. We have talked about media in the lab, and a general media is something that contains all the nutrients for microbes to grow. And we usually call them a general purpose media. We'll talk a little bit more about that. Media is when we're talking about more than one, when we're talking about only one, like tryptic soy agar, that we say is singular, and we call it medium. The plural is media. Medium can be liquid, meaning a broth, or it can be a solid, where it has something added to it to make it solid. Usually agar, but it could be something else like gelatin. In microbiology lab, we would give you a sterile media, and that means the media has been sterilized so it has no living microbes in it. And how we do that in the microbiology lab is we put the media in the autoclave before we give it to you. What you do is with that sterile media is you inoculate it with an inoculum. An inoculum is the microbes which are introduced into the media. The culture is then what we call the media while something is growing in it. So the microbes growing in or on a culture medium is called a culture. There's a very wide variety of media that are available to grow microbes in a microbiology lab. If you were attending a real lab, I could pull up a book of the different type of media. And each page in the book lists one media. And if you look at the size of that book, it's something like that thick. I don't know if you can make that out, but that's about 4 inches, at least 3 inches thick. And like I said, each page in that book is one medium. So there's a very wide variety of number of media that are available for growing microbes. The media should have everything that the microbe needs to grow. So the medium must provide an energy source, a carbon source, sources of nitrogen, sulfur, and phosphorus. The medium should supply trace elements, and if the organism requires it, organic growth factors. Now obviously the medium only needs to require this if the organism needs them. For example, a photoautotroph that fixes nitrogen from the media would not need an energy source if we're giving the cells light. It wouldn't need a carbon source because it's an autotroph. You can pull carbon from the air, and it fixes nitrogen from the air, so it wouldn't need a nitrogen source. So for a photoautotroph that fixes nitrogen, all you need to do is supply sulfur, phosphorus, and trace elements. And a photoautotroph isn't going to need organic growth factors because a photoautotroph will make all of its own growth factors. The trace elements would usually be supplied by the makeup of the media ingredients, or maybe just the water. So most bacteria, as well as people, get the trace elements they need simply from the liquids they consume, or maybe the food they consume. Any question about that? For example, for humans, we must get iodine as a trace element, and usually we get that from the food we consume or the liquids we consume. An organic growth factor that humans require is something like vitamin C. If humans don't get vitamin C in their diet, they will become sick and eventually die. Any question about that? Something like E. coli would need an energy source, and that would be something like glucose in the media. It would need a carbon source, like glucose in the media, a source of nitrogen, sulfur, and phosphorus in the media, trace elements from the water in the media, and E. coli, to my knowledge, doesn't require any organic growth factors. We can add agar to the media to make the media solid. Agar is a complex polysaccharide derived from seaweed, and we use it as a solidifying agent for culture media. You usually add agar to the media in a petri dish, but it can also be in a tube. Let me see if I can get this open. And we do have two types of tubes. Let me see if I can draw here. Not there. I might need help here. How do you draw? Let me see if I can get paint. I'm not seeing paint here. That's not going to be paint. Just a minute. There we go. So, the tube can be like this, where we have the media in a slant. And a tube can be a slant or a deep. A deep is where we have the media, sorry that's not a very good tube, and the media goes straight across. Oops, that's not very straight. You got the idea though. Did it again. Sorry, I can't draw a straight line. There we go. And it is possible you can have a tube where the media goes straight across, and then there's a slant as well. And so this is media, and this is media too. I guess what I should say is you could have it more like this, and then this is the media. Sorry, I'm not a very good artist, as you can clearly see. Any questions? So petri dishes, slants and deeps, all have agar. In a tube, if there is a liquid media, we call it a broth. Agar is generally not metabolized by microbes. And that's why it's the most commonly used thickening agent in microbiology for making a solid media. There are different types of culture media. Let me see if I can blow this up. There's chemically defined media, where we know the exact chemical composition of every molecule in the media. And the reason for that is that we add all of the molecules in the media. So here's a chemically defined media. It has 5 grams of glucose, 1 gram ammonium phosphate, 5 grams of sodium chloride, 0.2 grams of magnesium sulfate, 1 gram of potassium phosphate, and 1 liter of water. And we know all the molecules because we've added everything. Although it is true that the water if it's not distilled water, we'll be adding trace elements. Sometimes many growth factors may be required for the organism in order for the organism to grow. These are called fastidious organisms, organisms that require many growth factors. And this is a much more complex media where we're adding a whole bunch of organic growth factors to get something to grow. Chemically defined media are normally used for growing autotrophs because they're fairly easy to grow. And so we make the exact media for them to grow. A complex media is one that contains extracts of digestive yeast, meat, and plants. The yeast, meat, and plants provide vitamins and organic growth factors for the microbes to grow. The exact chemical composition of a complex media is not known. For example, in this media here containing peptone, beef extract, sodium chloride, agar, and water, we don't know all the molecules in the beef extract because different cows will be eating different things, and so different cows would have different molecules in them, which would mean that the complex media from one cow could be different from the complex media from another cow. Do you understand that? Okay, peptone is a partial protein digest, and the peptone is a primary energy, carbon, nitrogen, and sulfur source, similar to glucose, which supplies energy, carbon. I guess that's all glucose supplies. It doesn't supply nitrogen and sulfur. Complex media are used to grow heterotrophs of bacteria and fungi, and they require an organic carbon source. And they're a little bit more difficult to grow, heterotrophs, than autotrophs, meaning we have to supply a number of things like growth factors in order for the organism to grow. And the beef extract, as well as the peptone, can provide the things that the microbe needs to grow, like the growth factors. Any question about that? Another type of culture media is an anaerobic media. It's important for growing obligate anaerobes. And these organisms must avoid exposure to oxygen. Anaerobic media usually are also called reducing media because they have a chemical in them that is reducing, like sodium thioglycolate, that combines with and removes oxygen so that the oxygen becomes depleted in the sodium thioglycolate media. These media are usually heated before use to drive off the absorbed oxygen when the media was made. And usually what we do is we just stick it in the autoclave because you want a sterile media. And that heats the media and pulls off the oxygen. There's also selective media. This is a media that halts or inhibits the growth of a particular group of microorganisms, but does not affect the growth of another group of microorganisms. An example of a selective media is 7% sodium chloride media. most microbes will not grow in 7% sodium chloride. That's too high a salt concentration for most microbes to grow. And so 7% sodium chloride will select against those organisms and will select for organisms that will grow at 7% sodium chloride. Any question about that? An example of a media that will grow at 7%, not a media, a bacteria that will grow at 7% sodium chloride is Staphylococcus epidermidis, a bacteria that grows on your skin. Another media is a differential media. It provides differences between two or more species of bacteria. The differences are usually visible, and then you can see the differences between different species. The differences in the differential media are not related to how well the organism grows on the medium. So they may not grow equally well on the media, but the two species will appear visibly different. And so it makes it easy to distinguish colonies of different microbes. And an example of a differential media is blood agar plates. On blood agar plates, you can see differences in the hemolytic activity of the bacteria. You can see, let me blow this up, come on. You can see the growth in white here, and this is gamma hemolysis or no hemolysis, where the red blood cells of the agar come right up to touching the bacteria. No hemolysis. In alpha hemolysis, the bacteria shown in the alpha is white, and then there's a dark ring around the bacteria. The dark ring, which you can call greenish or dark, is where the red blood cells are partially hemolyzed by the bacteria. And so this is showing you alpha hemolysis, where there's darkening of the blood agar near the bacterial growth. And the most easy place to see it is inside the alpha. All this ring is darker. But you see the red here, and then there's a dark ring. Excuse me. There's a dark ring before the bacteria, and that's alpha hemolysis. With beta hemolysis, we have the bacteria in the beta here in white, and the beta hemolysis shows clear, and that's where all of the red blood cells have been lysed. And so here we have the red blood cells, and then there all the red blood cells are lysed around the bacteria shown in white. And inside the beta we also have all the red blood cells lysed. You'll notice around the beta hemolysis we have a ring, come on mouse, we have a ring of darkening, which is actually alpha hemolysis. And it's common to see alpha hemolysis surrounding beta hemolysis. Anyways, this is showing you differences in the hemolytic activity of 3 different bacteria. You can also have selective and differential media, a media that is both selective and differential. An example of that is ethene methylene blue, or EMB. It's selective against gram-positive bacteria, so they will not grow on the EMB media. And it's because eosine and methylene blue get stuck in the gram-positive cell walls, and the cell wall doesn't like that, and it prevents the bacteria from growing. Eosine methylene blue is also differential on the basis of the bacteria's ability to ferment the sugar lactose. So lactose is in EMB, and then a pH indicator is there. And if the bacteria, like E. coli, can ferment the sugar lactose, the bacteria will appear dark or metallic green. If the bacteria do not ferment the sugar lactose, then they will not be dark or metallic green. And here you can see the bacteria is like a white or off-white color, not fermenting the sugar lactose. And that's why this media is differential. It allows you to distinguish between a species that can ferment lactose and a species that cannot. Another type of culture media is an enrichment media, and an enrichment media encourages the growth of a certain set of bacteria. So therefore it is a selective media, but we call it an enrichment media because we use it to encourage the growth of a specific set of microbes. It increases the number of the desired microbe to a detectable level. This is particularly useful if we're looking for a microorganism when that microorganism is only a very small percentage of the population, which is often the case when we're looking for a specific species of microbe in the soil, another environmental microbe, or a human pathogen that we're obtaining from a fecal sample. Okay? So there's many bacteria in the soil or in a fecal sample, and we're looking for a specific one. We may need to enrich it by putting it in an enrichment media before we can detect it. For example, if the microbe is less than 1 in 100, if we were to streak it out on a plate and get 10 isolated colonies, we're unlikely to find the microbe that we are interested in because it's so rare in numbers. But if we were to grow it in an enrichment media and increase its numbers to 10%, then if we streak it out on a plate and get 10 isolated colonies, on average, one of those colonies will be the microbe of interest. So the point is that by enriching, we can bring the microbe to numbers where when we're streaking it out, we can actually isolate the bacteria of interest. Any question about any of that? An example of an enrichment media is when we're looking for a phenol-degrading bacteria from the soil. There will be very few phenol-degrading bacteria in the soil, so streaking it out would be pointless. But what we could do is take a sample from the soil and then put it in an enrichment media where the only carbon source available to the bacteria is phenol. and that will enrich for phenol degrading bacteria. So you transfer into a media containing phenol as the only carbon source, and then enrich the bacteria, and you can then take an aliquot of that and put it into another tube where phenol is the only carbon source to further enrich. And because only the phenol metabolizing bacteria will grow, we will increase the number of these bacteria so that we can streak it out and then get a good chance that we will obtain the bacteria of interest. Any question about any of that? This slide is showing you the different types of media we talked about: a chemically defined media, a complex media, a reducing media, a selective media, a differential media, and an enrichment media. It doesn't have selective and differential, but you should know that that is one of our culture medias, and it's both selective and differential. I've already talked about that. All right, let's talk a little bit more about streak plating, meaning streaking for colony isolation to obtain isolated colonies. What you want to do is take your loop containing bacteria of, well, I guess it doesn't matter, one or more species, and then streak it out, and then take your loop and put it in back to an incinerator to sterilize the loop of the bacteria, and then streak out quadrant 2, which you want to, when setting up quadrant 2, go through the bottom of quadrant 1. You do not want to go through the top of quadrant 1 because this will have the highest concentration of bacteria, but you do want to pick up a little bit of bacteria from quadrant 1 so that you make quadrant 2. And this drawing doesn't quite show it. What you really want to do is, at the very start, go through quadrant 1, which is showing you here, picking up cells and then streaking out quadrant 2, and then sterilize your loop in the Bacto incinerator, and then streak out quadrant 3 by passing it through, let me blow that up, by passing it through quadrant 2, 2 or 3 times. It looks like they did 1, 2, and then 3. And then streak it out. And as you can see in quadrant 3, we have isolated colonies forming. Colony streaking works very well when the organism to be isolated is present in large numbers. So the red colony here is present in large numbers. We can easily get isolated colonies of the red colony. We have another bacteria here. Let me see if I can blow this up. And that's the yellow colonies. And as you can see, the yellow colonies are at lower numbers than the red colonies. And so we're not getting isolated colonies of the yellow species. And so for that, we would have to streak it out from yellow, and we'd probably pick up red because there's no real isolated colonies of yellow. We'd probably pick up some red. So we'd have to streak it out again And even then, that wouldn't be a pure culture because this colony isolation would be the first time we get isolated yellow colonies. You'd want to pick from an isolated colony and then streak it out again, going through 2 colony isolation events where the yellow colonies are isolated 2 times before we call it a pure culture. And in this case, we'd have to streak it out 3 times to get a pure culture, because the yellow colonies, the first time, it's not an isolated colony. Everyone understand that? I think that was a quiz question earlier, or no, a lab question earlier. All right, let me move that up a little, shrink the size of that. About there. All right. When we're talking about the bacterial growth, remember we're talking about an increase in number, not the cell size. You should realize that bacteria can grow by 3 different means. We have talked about binary fission before, and that's where the bacteria cell simply splits down the middle and divides into 2. That we call binary fission. Most bacteria grow by binary fission. A few bacteria grow by budding, where the parent cell puts off a little bud at one end of the cell, and then the bacteria DNA divide, meaning copy, and then divide, and one copy is put into the bud. The other copy is left in the parent cell, and then the bud will slowly grow with time, and it will eventually separate from the parent when the bud is almost the same size, has grown to the same size as the parent cell. So that's how budding happens. There are a few bacteria that can also reproduce by a reproductive spore. Let me see if I can open paint again. We have a parent cell, which is big and usually elongated, and it puts off a number of buds on the edge of the parent cell. I'm trying to draw a 4 here. The point of the bud on the edge is that the bud can be broken off like by an animal passing by or by the wind, and can go elsewhere and then start growing the bacteria elsewhere. And this is a reproductive spore where one cell, the parent cell, produces more than one spore. And this is reproduction. Remember, I mentioned that an endospore is not reproduction because one parent cell produces one endospore, and then the parent cell dies. And so that's why endospore is not reproduction. With reproductive spores, one parent cell makes several reproductive spores. So that is reproduction. Any question about any of that? There's only a few bacteria that can reproduce by reproductive spores, and they tend to be filamentous bacteria. Any question about the different way that bacteria can grow? If not, let's take a look at binary fission, a little bit greater detail. The cell does elongate from when it first divided, so it does grow in size. And then the DNA replicates, why we have 2 copies here. And then the DNA moves to each side of the cell. And then the cell literally forms a cell wall in a plasma membrane in between the 2 sides of the cell. And the cell divides straight down the middle. And that cell wall and cell membrane grows right down the middle of the cell until we get 2 separate cells, and then they can separate, and that we have 1 cell dividing by binary fission into 2 cells. And here's an actual electron microscopic picture of this process of binary fission. Cells can also divide by budding, where one parent cell puts out a bud, and that bud can grow with time, as well as the fact that the DNA divides and 1 copy of the DNA is put into the bud. Let me see if I can get this to work. It looks like I need to push the, oh, there it goes. So there's the cell putting out a bud. Let's see if I go down, there it is. Putting out the bud is growing. And you can actually see the DNA starting to divide here. And right now the DNA is moving into the bud. This cell now separates from the parent cell, and it's close to the size of the parent cell. And there the parent cell is putting out another bud, and then the other cell is putting out another bud right there. So that is budding. Some bacteria can grow by budding. Any question about any of that? All right. So bacteria can grow by binary fission, by budding, and a few can grow by reproductive spores. When we're talking about the growth of bacteria cultures, it's useful to talk about the generation time. The generation time is the time required for a cell to divide into 2. This is also the same as the population generation time. So the time for 1 cell to divide into 2 is the same time for the population to double in size. The generation time varies considerably between species. Some bacteria can double as fast as 20 minutes. For example, E. coli can, at its fastest, double in 20 minutes. Most bacteria that we grow in the lab double between 1 and every 3 hours. Mycobacterium leprae, the bacterium that causes leprosy, doubles once every 10 days. So it grows much slower And generally speaking, the mycobacterium have a slower generation time because they have mycolic acid in the cell wall, and it takes the bacteria a long time to make that mycolic acid. Now, when we state the generation time is 20 minutes, it's understood that we are saying the generation time is 20 minutes per generation. Everybody got that? So when we're talking about a generation time, it's the time it takes for the generation to double, or the time it takes for one cell to double into 2 cells. And it's understood that that time, unit of time, is per generation. Students have a hard time with that, maybe because we're thinking about miles per hour, how we drive. But the generation time isn't that way. It's the time per generation. Okay, everybody see that? I'm getting no comments, so we'll move on. Shrink that a little.
Yes.
I heard something briefly. What was that?
Oh, I was just saying yes, I could see it. I just wanted to acknowledge.
Okay, thank you. When we plot the generation time, we use a semi-log plot. And it's not a log plot because the generation is linear, and that's the number of generations we're looking at. If we were to use the number of cells and not use a log plot, we'd get a graph looking like this, where one cell splits into 2, 2 cells split into 4, 4 cells split into 8, 8 cells split into 16, 16 cells split into 32, and the point of all that is you're getting a whole bunch of points all in this side of the graph. And then the points spread out and climb very rapidly. And then the next point here will be someplace on the ceiling of my apartment roof, because the graph grows very rapidly here. And that's what happens if you use a linear plot, meaning normal numbers. On the other hand, you'll also note that the curve is not very simple, and you'd have to explain it by something like y equals mx plus mx squared plus b, meaning it's not a simple equation to explain this curve. If, on the other hand, you plot it as a semi-log plot, where you plot the number of bacteria as the log numbers and the generation time as the actual generation number, and that's why it's a semi-log and not a log plot, you will get a curve like this, which you'll notice is a linear curve. It has a very simple mathematical equation, y equals mx plus b, and then the points in this region of the curve are spread out, and then the next one here will probably be at about here, so it won't be on the roof that we could actually graph the next point. We have to extend the graph a little bit, but we could graph the next point. So it spreads out the points in this region of the graph, and then we can get another point on the graph, which we cannot get on the non-semi-log plot. And for this reason, microbiologists like to use semi-log plots. Any question about that? And that's 3 reasons for using it. The points here are spread out. The equation is simple to explain it. And then we can get an additional point on the graph that you can't get if you don't use a semi-log plot. When we're talking about the phases of growth, almost all populations follow this growth curve. And certainly all bacterial populations follow this growth curve. When we inoculate a liquid media from a culture that's been growing in a solid media, we normally have a lag phase where the organism does not initially grow. And why that's the case is because the bacteria are making the enzymes they need to grow in this new environment. And it's a new environment because we inoculated from a solid media. We now are growing them in a liquid media. Once the organisms make the enzymes they need to grow in this media, they then start growing. So we leave the lag phase where the organism isn't growing and then enter the log phase or the exponential growth phase where the bacteria are growing rapidly. The reason the bacteria are growing rapidly is the bacteria are low in numbers, the nutrients are high in numbers, the waste products are extremely low in numbers, and there's lots of room for the bacteria to grow. So the bacteria grow very rapidly, and that is the log or exponential growth phase. But that phase doesn't last forever. As the bacteria population grow, the growth will start to slow, meaning the S curve right there. And why the bacteria slow is because the nutrients are becoming limited, the waste products are building up, the space is becoming limited, and the environment may be deteriorating from all of the bacteria growing. So the growth of the bacteria slows. Eventually the bacteria will slow and we'll have the stationary phase where the bacteria growing equals the bacteria dying. And so the population number of bacteria remains the same. The lucky bacteria will be growing because they will be in an environment where there are some nutrients for the bacteria to grow. The waste products are not excessive, so the bacteria can grow, and there's enough space for the bacteria to grow, as well as the environment hasn't been detrimental for the bacteria to grow. The unlucky cells will not be in such a lucky environment, and there will be too much waste around them, not enough nutrients around them, and not enough space around them, and they will die. Those are the unlucky cells. In the stationary phase, the number of cells reproducing equals the number of cells dying, but that will not continue. Eventually, the population will enter the death or decline phase, where the number of cells dying is greater than the number of cells growing. and then the population number declines. Now, late in the death or decline phase, we will not have any cells reproducing. We will only have cells dying. And that's because the nutrients are all used up, there's lots of waste products around, and space is limited. And so the cells are not reproducing, they will eventually all die. How long it takes for the cells to die depends on the species. What do you call it? A finicky and sensitive cell can die fairly quickly, maybe within a couple of days, certainly within a few weeks. But if the organism produces endospores, this could take years, decades, millennia. Everyone understand that? Anyways, these are the 4 phases of growth: a lag phase, a log phase, a stationary phase, and then the death or decline phase. All right, the next slide talks about the microbial growth, and you're going to talk about that in your in-class activity, so I'm not going to talk about it. Let's move on. Oh, well, actually all of the slides are talking about this, so let's just go ahead and talk about them briefly. I've only got 2 more minutes, and when we end, we will just end it. There are different methods for measuring the growth of microbial populations. There are direct methods and indirect methods. The direct methods include the viable plate count, the filtration method, the most probable number method, and then the direct microscopic count. The indirect methods, meaning we're counting the cells indirectly, include turbidity, the metabolic activity, and the dry weight. I've used all of these methods to get a count of my cells, except for the most probable number. I've never used the most probable number to get an estimate of the number of cells I had. All right, any question about any of that? The next slides discuss it, and you'll be talking about these different methods in your worksheet for this lesson. Are there any questions? If there's no questions, I'm going to end the session, and I'll see you at 6:30 for the lab.